in Switzerland we used to work on the Franche-Montane horse breed, so it's the last native horse breed there. So for Swiss people it's quite important to work on that horse breed. And we also, what we're focusing there is also genetic diversity. So when it comes to horse breed, I think this is one of the major questions we are going to focus. And what we are currently using, so I put some lines here, is like the contemporary biology at the moment is using high density SNP genotype information to a certain genetic diversity within populations. So you have to imagine, so previously or previous studies, they were only limited to macrosatellite data or just pedigree data. So there's a big advance in that technology we can use, and that's available at the moment. And just for you to better understand what's even is a SNP. So it represents a single pair change in the genome, and for students I always use this text marker, and for a good comparison it just highlights you in the genome really important positions. So you have to compare that if you read in a book, so the important text you highlight with that marker. And this is also the reason SNPs are referred as genetic markers. So what's going on in the community? So there we're talking about different SNP arrays that are available for horses, and they can contain different numbers of SNPs. And for horses nowadays there are like 2 different types of SNP chips available. So I put 2 SNP chip markers here. So there's one from Illumina. So on this one you find 54,000 SNPs. And more recently, so they released the 700K SNP chip, and this is FE matrix. And here you can already see the important players in that business. So this is Illumina and FE matrix. They are like the drivers in that technology. Professor Brame already mentioned in the morning, so there's also whole genome sequencing platforms that are becoming available. So this is still a big difference in the pricing for, if we're talking about SNP array genotyping, we're talking about 200, 300 euros. When it comes to sequencing, whole genome sequencing, we're talking about 2,000 euros, something around that. But it's becoming cost effective, so there are lots of genomes fully sequenced out there in the world. And Professor Brame already showed this Twilight thoroughbred mare, because this is very important for horse geneticists. So this is the reference genome, and all we refer to is connected to that mare. So here just a brief summary what you actually can do with this SNP genotypes. So you can perform genome-wide association studies. So what does that mean? So genome-wide association, so that means you have like a phenotype record, and then you have like genetic informations, and then you want to combine that, and you just look into the genome if there is like a special place in the genome that is responsible for that trait. And we did such an analysis for like prognatism in horses. They recorded in the Franche-Montane horses if the horse is affected by prognatism or not. So this was like a case control study. Another possibility would be selection signatures. Selection signatures you will perform if you don't have any phenotype available, and then you would just compare the informations within a breed or between breeds, and we conducted such an analysis in Shetland ponies to look in body size. And there we found a region that was associated with body size on chromosome 1. Then what's important for us is like genetic diversity analysis. That was already conducted on a really big horse data sets involving numerous different breeds. That was a paper in, I think, 2014 by Peterson et al. And here you can already see such a network tree, and here you can see how the different horses are related. So let's go to our aims and objectives. So what we want to address because I already told you there are now different genotype platforms around. So we want to evaluate the genetic diversity using different SNP densities and see if they are any different. Then we also want to assess the population structure of the horse breeds using the common applied programs. And what we also want to do, that we developed in-house is a novel three-step approach, and we also want to apply that. And in future, we also want to look into selection signatures of our sampled horses, also using common applied methods and strategies. Okay, let's come to our data set. So what we have collected so far. So this is our preliminary data set. So we are in discussions with other research groups to extend this data set to make the analysis even more attractive to the people in the community. So what we have here is like Achaltekina. This is coming from the University in Bern, because they were genotyping these breeds because they were investigating the genetic background of the naked foal syndrome that's apparent in this breed. Then, of course, we have the Franche-Montane horses. So this is coming from our stud farm. Then Thomas provided some data, including the Lipizzan horses. And besides that, we have Haflinger, Norica, and Thomas was also collecting with a colleague Shager Arabians. And from another study investigating Aereo in Switzerland, we also got some warm blood samples that they were mainly collected in Switzerland. And you also see there are two breeds with some stars, and this was warm blood and Franche-Montane horses. And here we also had some information on whole genome sequencing, from full genome sequencing. That's the outline of the methods I briefly mentioned before. This is this new three-step approach we applied also on this data. And this starts with the identification of key contributors. So based on that method, we are able to identify horses that had a really high impact on the formation of the population. And we combine that method with admixture analysis, so we can also identify if there are highly admixed individuals that had a great impact on the population. And the third step, this is a network visualization. And I will talk about that in detail later. And altogether, this provides you a high-resolution population structure analysis. The first thing we thought about is how these different platforms, how their SNPs do overlap. And what we see here is like that we fully cover the 50K SNP chip information except one single SNP. So we find that in our full sequence data, and I think there's also a pretty fair overlap between the sequence data and the 700K data. So why is this important? This tells us this is really a good base that we can merge data together, so we can combine data from full genome sequencing with high density genotyping. Here already some first results on the structure analysis. So here you commonly use a principal component plot, and there you plot the first components versus the second component. And then you already see how your different horses are related to each other. And what we see here, that on the first component, already the Lipizzan horses, they got differentiated from all the other horses. Whilst on the second component, you see the variation between the other breeds. And what we also see here is like the geographic relatedness between the different horse breeds. So you see that there is like the Haflinger horse breed and the Norica horse breed, so they are in a proximity. Then what's also surprising me was there was really close relationship between the Shager Arabians and the Achaltekina. And what we also see, there are some Franche-Montane horses going towards the Shaggy Arabian and the Haltekina cluster. Right? But we know that there was some crossbreeding in the breeding history of the Franche-Montane horses, so with warm blood. And what's also interesting to say, for example, for the warm blood horses, is that there might be also differentiation when we see there's like the sequenced horses and the genotyped horses. This we still have to further investigate. What else we can do based on this outcome of principal component analysis? We can also determine the optimal number of clusters. And here we have the top value around 8. So optimally, this is a good fit for our data. Other approach you can apply to investigate population structures is admixture. So maybe some of you know the older version of that kind of analysis, that was the program structure. And here you perform a clustering along a series of number of clusters. So you start with K2, and here I put the K2 till K8, and here you can also see how the different horses were clustered along the series of the number of clusters. And here again, so this also confirms the principal component analysis results. So the Lipizzan horses cluster out quite early, so got differentiated from the others. Then the Norica horses clustering out, followed by the Haflinger, then the Franche-Montane horses, and then in the Franche-Montane horses you already can see they are like admixed individuals that show an admixture with the warm blood, what we know. So this is confirming also the knowledge or the breeding history we know from the breed. And what you can also see is that Shaggy Arabians and Haltekina, that they stay in one single cluster. And more interesting is the result in K7. So you see that there might be a substructure in the Norica breed. And this is quite surprising because at K equals 8, this substructure disappears, and we get more substructure in the Lipizzan horse samples. So there will be 3 subclusters in that breed. And what we also have seen is that based on that method, you can also determine the optimal number of K. So this would be identified by the lowest value. And what we say here is that admixture runs into a problem to find an optimal number of clusters, because already at 15 there's the lowest and it's still going down. So it was actually suggesting there are like 15 different clusters. And this was what I mentioned before. This is now this high-resolution network. And here again, here for example, we get more information according to Shaggy Arabians. So we see there's some substructure here. And what we also see is we have some really highly important horses in the Lipizzan breeds, whilst in the other breeds we do not have such dominant horses there. So they are like more unrelated, and that's the reason we don't see such a picture in the other breeds. So let's go into more detail with the Lipizzan horses. I think that's the reason you're here. So let's talk about the Lipizzan network. And here I just put the names on the horses that had a really high impact. But keep in mind, this is just a sample from PIBA, so from the PIBA stud farm. So I think there are ideas to extend this population sample, but what you can already see that the highly impact of Pluto Briosa, Favora Superbia, and Conversano Cesano. So this was the stallion on the booklet they were publishing. So maybe that's the reason now people prefer this stallion. And what we also see here is, because I told you that admixture identified some substructuring in this sample set, this is also connected to this family relationships. What we see here is that there are different family exists, and that some of the stallions had a really great impact on that population sample. So for the conclusion, next steps, what we can say is that genome-wide SNPs are a very useful tool to ascertain fine-scale population structures within horses. And the other thing what we want to point out is that the combination of currently independent applied methods into this three-step approach allows the identification of population structures. And very important for us is this is without the need of any individual ancestry. So this is completely independent of that. And why is this so important? Because for us, for all the subsequent analysis that following for interpretation of genetic diversity and selection signature analysis, this is very useful for the data preparation. So to set up to select the best data for the specific analysis. So, and what we think based on this first-hand results, we want to move forward to selection signature analysis, what we hope that we'll perform in future. So these were just the first preliminary results are provided here, and this work will be continued. And so this is the end of my presentation, and I'm happy to take any questions. Thanks.